Good evening. This is Carly for Cell Biology Lab. This is my protein presentation. My protein that I got was actin, and there is so much to learn about actin. There's so much information about actin. It's so hard to condense. And so um I try to condense it as much as possible because if we had too much of it, too much information, then we'd never get through these slides. All right, so we're gonna move forward. We're gonna talk about general information on actin before we talk about any kind of functions. We're gonna talk about the functions, diseases caused by actin, all sorts of stuff about actin. But first, we're gonna learn about actin in general. It is the most abundant protein in the eukaryotic cell. It accounts for 5% of the total protein in an animal cell. It is assembled into filaments, F-actin, which are a bunch of G-actin, which we're going to see further on, and G-actin are monomers, and the filaments are comprised of G-actin that are wrapped around each other, and if you see on the right, that is G-actin coming together to be F-actin, so G-actin, and then if you look right here, they're starting to form together, and this is F-actin. It starts to swirl around each other like you'd see curly hair, and it comes together and forms these F-actin filaments. And about half of the actin in a cell is comprised into F-actin or actin filaments, and the other half remains as monomers inside of the cytosol. Actin participates in more protein-protein interactions than any other protein, so it uses other proteins to do its duties. It's the only one that uses some of these other proteins that sometimes could be totally irrelevant, but actin has many duties, and without actin, our body would not function the way it does. Critical to many cellular functions such as mobility and maintenance of cell shape, along with the polarity to the regulation of transcription. So without actin, we wouldn't be able to transcribe genes into other genes, or we wouldn't be able to have offspring, and we wouldn't be able to move without actin. We wouldn't be able to move our muscles. We wouldn't be able to divide cells in cytokinesis. Actin has a lot of duties. Functions of actin in the cell. Actin monomers come together to form filaments, as previously stated. Without these filaments, animal cells could not move, cells could not divide to make daughter cells. With the help of actin binding proteins, actin regulates cell structure and activity and gives rise to actin bundles and networks. These actin bundles and networks that we're going to learn about a little bit later, they are what drives cells to form tissues or form rings that will split a cell into two daughter cells. Actin plays crucial roles in cell adhesions, cell contractibility and mobility, signal transduction, transcription and regulation of RNA and DNA. It's basically transcription of genes, and cytokinesis. We see actin in cell adhesions and morphology. Actin is key to integrating cells into tissues. The cytoskeleton is crucial to cell-cell junctions. So basically, the cytoskeleton gives a shape, it gives a form, it is the structure within the cell. And it forms the adhesions between 2 or 3 cells and forms tissues which turn into organs or bodily systems. All morphogenic rearrangements rely on movement and shape changes made by the cells within the tissue. These changes rely on actin and allow changes in overall tissue architecture. So actin is very important in determining cell shape, and cell shape determines what type of tissues these cells are going to make. So muscle cells or brain cells or cells that make veins or cells that form nerves that send signals throughout from the brain to your foot. Tissues made by these adhesions result in bodily structures such as muscles and organs. And as we see to our right, we look at that thin actin thin filament. We see right here. I don't know if I'm highlighting it quite well. Let's see. Let's see if I can. Actin thin filament. And my computer doesn't like me highlighting that. But anyway, the actin thin filament, it forms with myosin to make muscles inside of the body. Cell contractibility and mobility. Actin is the main determinant of cellular mechanics. A cell cannot divide or move without actin. These mechanics are determined by actin filaments' densities and lengths as well as proteins cross-linking them. Actin filaments require myosin to form the basis of tissue movements. The continuous turnover of cortical filaments enables cells to quickly respond to stimuli such as signals from the brain to make muscles move. So your brain sends a signal to your foot to move. And this is actin. Without the constant turnover of your actin within your body moving, your brain wouldn't be able to tell your foot so fast that you don't even realize like you're telling your foot to move. It wouldn't be able to do that. We would have to think about it, and then we'd have to tell our foot to move. We don't have to tell our foot to move because our brain tells us our foot to move when we want to move it. And thanks to actin, that makes it fast. Actin plays a part in skeletal, cardiac, and smooth muscle contractions and architecture as well as non-muscular cells. So we wouldn't be able to digest food to make energy or anything like that without actin. Muscle cells within the body are packed with actin filaments with positive and negative ends that are arranged in the bundles. Actin bundles contract as myosin and motor proteins move towards the plus ends of the filaments. So as we see in the right, we have plus ends that move together, or plus ends and negative ends that move together to contract, and the filaments slide, and that's what like when we see, as we see on the next slide in cytokinesis, the cells move together and they turn one cell into two daughter cells. So that's the easiest way I think of actin. Actin is the reason why a cell splits. It forms that ring that splits a cell into two, which is so amazing to me. Such little things can do so much to our bodies. Actin also plays a role in signal transduction, which is where cells respond to stimuli, outside stimuli, these frequently include changes in cell movement and shape. So actin changes the shape, it moves the cell, and it's doing this in response to some type of stimuli, outside stimuli. And actin responds to that stimuli for the cell. Signal transduction is the process in which chemical or physical signal is transmitted through the cell that results in a cellular response. Growth-induced changes in cells play a crucial role in processes such as wound healing and embryonic development. This is overseen by the actin cytoskeleton. The assembly and disassembly of actin filaments with plasma membrane characterize the response of cells. DNA, RNA, genes, actin plays a huge role in transcription and regulation, and this is one of the most controversial topics that I found. It's just crazy how many different types of theories that people have about DNA and what actin has to do in DNA. But DNA is so hard to look at, and seeing actin inside DNA, trying to work with DNA is hard to hard to see. So, and it's hard to experiment with. It's hard to look through a microscope. There's a lot of hard, it's too small. It's so small, and so maybe one day science will get good enough to where we can see everything inside of a DNA and plus more. what's inside of that DNA, what's inside of those amino acids. It's insane the amount of things we don't know about this protein, and it's insane the amount of information there already is about it. But anyway, transcription and regulation of RNA. Actin binds transcription factors and determines their cellular localization. Actin has been proven to regulate gene expression by placing transcriptional regulators within the cell. Actin binds directly within RNA polymers and is required for transcriptional activity. Actin aids in transferring mRNA from the nucleus to the cytoplasm. Actin regulates chromatin, the material used in transcription of genes. So, I think as previously stated, we did the signal transduction. Actin tells the cell where it's going to split and which DNA is going to go into each one of those daughter cells. It transcribes those genes, puts them into two daughter cells, and splits it. And that's the easiest way to look at this, and that's the easiest way to think about actin and its duties, or how it functions this way. This next slide, we see cytokinesis. Now, cytokinesis is kind of like all three of these previously stated functions put together. It divides the cell with the contractile ring that pinches a cell in half, driven by F-actin and cell division. It also signals where the division is going to happen, and it carries both daughter cells, transcribes genes into both of the daughter cells. So whichever genes go into which daughter cells is driven by the actin. Functions of actin in an organism. So we're looking on a bigger scale now. What does actin do in our bodies? Not in just our little cells within our bodies, our whole body. Actin is the main component of movement within the body. Without actin, we would not be able to move. It has one of the biggest roles in the body by being the main component of cell-cell junctions and movement within muscle tissue. Muscles control the whole bodies from functions of organs to the muscles and tendons adhesions. So muscles and tendons and bones, that adhesion, if it didn't happen, or if it didn't have actin, it wouldn't happen. And our muscles wouldn't move. We wouldn't even have muscles. It would just be flappy. We wouldn't have them because there wouldn't be actin filaments holding them together. This is the protein that holds your muscles together. It builds your muscles. Muscles contract to move the body and control organs. So the contractibility of the actin filaments is what contracts muscles and moves the body. So now we're going to get to slide 10, levels of structure within actin. This is primary, secondary, tertiary, and quaternary structures. Our primary structure of actin is characterized by its first 6 amino acids. And actin is made up of over 400 amino acids, and we'll never know every single one of those acids. There's too many to name. But the first 6 we can name: methionine, cytosine, cytine, aspartic acid, asparagine, and valine. This primary structure is permanent and stable. It's never going to break. Secondary structures, secondary, tertiary, and quaternary, all can change. They can all unwind because they are unstable, and they can be broken because of the hydrogen bonding. It's not a permanent bond. Secondary structure is characterized by the local folds that the peptide chain develops: A helices and beta sheets. Alpha helices and beta sheets. This is what secondary structure is. And then tertiary structures, the structure of the G-actin itself, monomers, the full monomer, with the beta sheets, the alpha helices, the primary structure of the 6 amino acids and all of its covalent bonds. That is tertiary structure, everything put together. And the quaternary structure is the same thing, but just with the F-actin. It's F-actin, which is made up of 2 G-actin fibrous elongated strands that are wound around each other. These are breakable and can be unwound because of their hydrogen bonding. Genetic diseases. So, we're going to look at genetic diseases, and this is one of our last slides, so I'm not going to bore you for too much longer. A slight change of mutation of actin, which is a change in the in the actin shape, what shape the actin decides to make the cell. Some type of amino acid change. This is a mutation of actin within DNA, and it can result in many diseases. Congenital myopathy is one of them. It's a genetically determined muscle disorder present at birth. This would be scoliosis, ALS, things that your body can't control. It's your muscles deteriorating or your bone marrow not being strong enough, anything like that. And this is the saddest slide to me, is the fact that actin has so much of a presence in your body that it even can hurt you. It can be life-threatening or benign, and results of malfunctioning actin in the cell-to-cell junctions. So when cells come together to make your muscles. Comprised immunities. These are cytoskeletal defects that cause immunodeficiencies without impacting development. This affects actin that are regulatory proteins within the immune system. So this is talking about your immune system not being up and ready to fight off diseases. Neurodegenerations. These diseases are affecting the brain. Actin proteins with the functions of signal transmittance and build brain tissues and connections can be mutated and affect the brain. This is why we have Alzheimer's disease, Parkinson's disease, ALS. This is why we have dementia. Dementia, it's your brain losing its connections. These connections are formed by actin filaments. And when those actin filaments are mutated, then you lose your ability to make memories, or your ability to remember things. Cancer spread. Lastly, cell migration governed by actin is required by many biological processes, including tissue repair and regeneration. When mutated, unregulated actin cytoskeleton can be transferred to other parts of the body, causing spread of malignant cells. So these malignant cells, when the actin has some type of mutation that forms a cancer that's malignant to the rest of your body, then it can be spread because actin filaments are sometimes your body doesn't regulate them. They're unregulated. So they just keep transferring to other parts of your body, and that's why cancer spreads. All right. Well, that completes my my presentation. I hope you have a wonderful night, and thank you for listening to me present.